Hi there, science fans, and those curious about medications or drugs you take and how they affect your physiology or how you feel. Dan Casciano here to present the third episode in a series describing the structure and function of the human cell. I am structuring these presentations to provide you a sense of the basic unit that makes up your body. Hopefully, this effort will give you some basic vocabulary and information that allows me to then transition to what I think is the basis for developing this channel. I wish to help those of you curious about how your body works, and more importantly, how we as senior citizens become more familiar with the drugs prescribed to relieve some malady we are experiencing or help us adjust to the aging process. In addition to these more formal presentations that are to a certain extent academic, I've decided to include presentations focusing on special topics that are in the news and try to explain them to you in plain language. One such topic has to do with understanding one's microbiome. Most of you are familiar with the gut or intestinal microbiome and how probiotics may be beneficial to the health of that microbiome. I will spend some time introducing the concept of maintaining healthy microbiomes and describe how drugs, nutrition, and other activities in our daily lives can influence the health of our microbiomes. There are other important microbiomes that reside in different niches of our body that are essential to our health, and I will describe some of those to you as well. In order for this series to be successful, you must help me enhance the quality of the topics and presentations by submitting your comments and suggestions below. In order to do this, please click the subscribe button and ring the bell to be informed of future videos. Thank you for your attention. This is Dan Casciano, signing off. In the previous episodes in this series, I discussed the structure of a typical human cell and some of its internal organelles, as well as the structure and function of the nucleus of a typical human cell. In the next several episodes, I will focus on the structure and function of human DNA. In this episode, I will discuss the structure of DNA in relatively simple terms, followed by several episodes in this series discussing the complex function of the DNA. DNA is a polymer consisting of monomers. An analogy would be a brick house and individual bricks. The individual bricks are monomers, such when placed together in a specific way, form a house, which is the polymer. In DNA, the monomers are called deoxyribonucleotides. And like the house's architectural structure, the DNA has a helical structure. Each DNA strand or chain results in a double helical structure. DNA is not only found in the nucleus in the form of a chromosome, but is also found in the energy organelles called mitochondria. The DNA is like a blueprint that contains the instructions for making an exact copy of itself. This process is called replication, and also contains the information for the formation of an RNA message. This process is called transcription. This message eventually is translated to cellular proteins that are necessary for the function and survival of the cell. DNA is a nucleic acid, a polymer, polynucleotides, or long chain of a series of building blocks, monomers, called nucleotides. Each nucleotide consists of a nitrogen-containing aromatic base. It is attached to a 5-carbon sugar called a pentose, and those two molecules are attached to a phosphate group. The nitrogenous bases are the letters of the alphabet that instruct replication, transcription, and translation that I discussed in a previous slide. The 4 bases are adenine, thymine, guanine, and cytosine. These 4 letters, or bases, serve as the primary building blocks of DNA. This slide depicts the adenine, thymine, guanine, cytosine pairing. Adenine always pairs with thymine. Guanine always pairs with cytosine. Earlier, I showed a helical structure of the DNA, If that helix is flattened out into this ladder representation, one can more easily see the phosphate sugar backbone and the pairing between the nitrogenous bases. As you can see in this representation, A is always pairing with T and G is always pairing with C. The 3D structure of DNA is depicted on this side of the slide. Once again, we have the double helix, each strand wound around a central axis. This central axis is wound about by a sugar phosphate backbone, and within each of the strands there's bases that pair with each other, forming the obligatory pairing of thymine with adenine and guanine with cytosine. Once again, if we flatten this structure, we can see here in this cartoon that the backbone is oriented in opposite directions. One strand is oriented in the 5' to 3' direction, and the other is in the opposite direction. We can also see once again the ATGC obligatory base pairing. This slide indicates a linear representation of the polynucleotide chains. Once again, we observe that each of the chains are oriented in opposite direction to its complementary partner. We also see the required pairing of the nitrogenous bases on each of the complementary strands. A always pairs with T, and G always pairs with C. If we imagine that this segment represents a small gene responsible for the formation of the imaginary protein X, we observe that the sequence of the DNA alphabet necessary for its transcription and translation is responsible for the normal formation of the protein X. If this sequence is altered, a faulty or inactive protein will be formed. And if this protein is essential for the survival of the cell may die. More of this in the presentations on the function of DNA. This next slide shows two cartoons representing how DNA coils and is contained within the nucleus. The cartoon on the left shows the DNA in this double helical conformation, and it resides in a chromosome. In every normal human cell, there are 23 pairs of chromosomes responsible for how we look, how we behave, etc. The cartoon on the right shows the same information in greater detail. This tightly coiled DNA allows 3 billion base pairs contained in the 23 pairs of chromosomes, and it fits in the space of 6 times 10 to the minus 6 meters. That is 6 zeros before the decimal point. So it fits into a very small space. Each human body contains approximately 10 billion miles of DNA. In the next episodes, we will spend some time on the function of the DNA, that is, replication, transcription, and translation. In this particular slide, I've chosen a photo of Watson and Crick, Jim Watson and Francis Crick. These are the two scientists who deciphered the structure of the DNA as depicted in this particular photograph. They received the Nobel Prize in 1953. See you next time for episode 4